Welcome to the 8th lecture in this module. In the last lecture we talked about the ability of cannabis-based medicines to alleviate some of the symptoms associated with cancer or its treatment. And in this lecture we're going to look at what is the evidence that actually there might be an anti-cancer effect of cannabinoids or cannabis-based medicines, and do they have an ability to actually reduce tumour growth and tumour size. So why do I need to know this? If you go on the internet right now, the internet is rife with stories that cannabis cures cancer. And I think if you are a patient or a carer of somebody who has cancer, it might be quite easy to believe, looking at all these articles and news stories and anecdotes of individuals who've had very good success with cannabis-based medicines. It might be quite easy to believe that cannabis could cure cancer. And many users of cannabis-based medicines, although they may use their cannabis for symptom relief, I think a lot will admit that deep down that they're hoping that there will be some impact on their disease progression as well as symptom alleviation. But what is the evidence? I mean, this is really what I've been trying to cover in this whole module. What is the scientific and clinical evidence that might be behind this? Is there any truth in these stories? And for this, I'm actually going to go about things the opposite way around than I have in other lectures. I'm going to start with the preclinical evidence. Normally I've talked about what clinical evidence we have, and then I've talked about what science we have to back that up and how we know these compounds work. But actually I'm going to do it the opposite way around. And that's really because we know a lot more about the anti-cancer effects of cannabinoids in the preclinical arena. So we have a lot more science in this area than we do have supporting clinical data. So I want to start off firstly by saying there is very strong preclinical evidence. So remember, that's evidence from cells and animals that cannabinoids have anti-tumoral properties. So when you read those headlines, biochemist finds that THC cures cancer, there is truth in that. So when these experiments are done in laboratory settings, cannabinoids are very good as anti-cancer agents. And there are really 5 areas that we have a lot of information on how cannabinoids work in cancer. So the first is that they can actually kill cancer cells. So these cells that are growing abnormally within the body, cannabinoids are able to kill them, to cause cell death within cancer cells, specifically within the cancer cells and not healthy cells. So they seem to be a lot more effective as cytotoxic agents, which is the word meaning cell killer, in cancer cells than they are in regular cells. It's also been shown that cannabinoids can slow cancer cell growth. So the ability of these cancer cells to grow and divide and grow and divide and grow and divide is slowed down by the presence of a cannabinoid. So we call that anti-proliferative. So proliferation is a word for meaning cell division. And so in preclinical experiments, cannabinoids have been shown to be anti-proliferative, to really slow down that growth and division of cancer cells. So those features are when we look at cells in isolation, in petri dishes, in cell culture. But there is studies in animal models where tumours are induced by injecting cancer cells into the animal, and then that cancer grows and produces a tumour in the animal. And cannabinoid treatment in these animal models has been shown to reduce the size of the tumour or to prevent the tumour from growing in an animal model. Another aspect that cannabinoids have been shown to be beneficial in is the ability to stop new blood vessels from growing around a tumour. Now, a tumour needs feeding. As it grows and gets bigger in mass, it needs a blood supply to help it to grow. It needs feeding like any other cell. It needs oxygen and it needs nutrients, and it needs something to help it grow and divide. And so tumour cells normally send out signals to kind of promote new blood vessel growth around the tumour, which is called angiogenesis. And what this does is it feeds the tumour and builds a blood vessel network around it. But cannabinoids have been shown to limit that signal and to prevent the formation of these new blood vessels growing to the tumour, which is obviously very important in limiting its nutrient supply. And then finally, it's been shown that cannabinoids can limit the spread of cancer. So when a cancer starts in a particular location in the body, it can grow, and then some of the cells can migrate, can move to other areas of the body, which is called metastases. And then you can have tumours forming in other areas other than where they initially started. And we do have preclinical evidence to show that cannabinoids stop that from happening. So they limit that spread of tumour cells, and therefore they limit the development of new tumours or nodules growing in other areas of the animal other than where the cells were initially injected. So let's look at each of those elements in a little bit more detail now, because this is quite a well investigated area. So the scientific literature that supports this is really quite exhaustive. And so we can go into a little bit more detail on each of these elements. So I just want to say a little bit of some general information about the anti-cancer effects of cannabinoids, and starting first about which cannabinoids are effective. And the summary, the bottom line, is pretty much everything that's been tested so far, every type of cannabinoid that has been tested so far has been found to have anti-tumoral properties. Now, I'm not saying that they all necessarily do each of those 5 points that I've just highlighted, but they've all been found to have an anti-cancer effect, at least in some aspects of tumour progression, growth, division, and spreading. So just to break that down, it's been found that many of the phytochemicals are effective anti-cancer agents. The most research that we have is on THC and CBD, like in most other scientific arenas. They're the compounds that have been investigated for the longest. We know the most about them. We particularly know quite a lot about CBD. Part of that will be because from the early days, when it was realized that cannabinoids have anti-cancer effects, I think people focused on CBD because of the potential to maybe develop a medication that doesn't have the psychotropic side effects of THC, which to some patients are really not very tolerable. So I think a lot of research has very much focused on the anti-cancer effects of CBD, and we have a lot of preclinical evidence that CBD is effective in cancer models. And some of the other minor phytocannabinoids, we also have evidence that they work. So CBG, CBC, CBDV, CBN, THCV, and also we're beginning to learn a lot more about some of the other phytochemicals that come from the plant, like flavonoids, also can have an anti-cancer effect. So far, all of the, and there's many phytochemicals that have been shown to do this in laboratory studies. And people have also looked at trying to selectively target either the CB1 or the CB2 receptor, and molecules that have been designed to activate either one or the other or both are all effective anti-cancer agents. Endocannabinoids have also been shown, so direct application of endocannabinoids such as anandamide and 2-AG, which are the endocannabinoids that activate the CB1 and CB2 receptor, but also some of the other endocannabinoid-like molecules like oleol ethanolamide and palmitoyl ethanolamide also have anti-cancer effects. And we can also modulate the endocannabinoid system to upregulate endocannabinoid tone. So we can do that with a couple of different strategies. We can use enzyme inhibitors, so we can block the Fa and the Magel enzyme that normally break these compounds down. So that means that there's more of them around. Or we can block their transporters. So FABP5 is fatty acid binding protein, and this is like a bus that binds endocannabinoids within the cell and moves them around cellular compartments, including to the enzymes for degradation. So if you stop the endocannabinoids being transported to their enzymes for degradation, then you can prevent them from being degraded. And all of these strategies increase the intracellular and the extracellular concentrations of endocannabinoids, and that means that they can activate their molecular targets like CB1 or CB2 or any of the other molecular targets that I've talked about before, and this can bring about an anti-cancer effect. So far, pretty much all types of cannabinoids are effective as anti-cancer or anti-tumoral agents, at least in preclinical studies. And which cancers work? Well, so far, again, almost every cancer type that has been investigated through these types of studies, cannabinoids have been found to be effective in. So that includes, but is not limited to, brain, colon, breast, ovarian, endometrial, prostate, lung, renal, thyroid, liver, gastric, skin, and blood cancers. So these are all cancers that we have preclinical evidence that cannabinoids work in. And again, I'm not saying that every cannabinoid works in every type of cancer, and actually a lot of the devil will be in the detail here, is that we may find, because every cancer is different, we may find that particular cannabinoids are going to be more beneficial in particular cancers. But I think that level of detail isn't really known at this stage, because people tend to focus on a particular cancer type because that's their speciality. But so far, cannabinoids have been shown to be effective in every cancer type that has been tested. So there's a lot of references for this, for what I'm presenting to you today, and you can go and look this up further. I haven't given specific references on each slide, but rather given you a selection of very good reviews that you can go to if you want to read about this in further detail. So let's start with this idea that cannabinoids kill cancer cells in vitro. This is a headline that you may have seen. And I wanted to first show you what we mean by in vitro, because if you're not a scientist, this might not mean very much to you. And these diagrams here on the right are, firstly, the one on the lower panel is a selection of cells grown on a petri dish. So you can see they're grown stuck onto a plastic bottom. They all grow next to each other in a sheet, so it's not in a tumor formation. It's not in a ball. They grow in a sheet on these plastic dishes, which the picture above shows you. They're grown in an incubator and they're kept in a solution, which is a little bit like plasma. We call it cell culture medium, and they're kept at body temperature in oxygen and carbon dioxide. So it's a fairly unphysiological situation. This is not how tumors grow in the body. And so when you read these types of experiments, you have to kind of have that in the back of your head, that this isn't a very physiological reenactment of what it would be like to actually try and kill a cancer cell if it was in a body. And the other major difference is that when we're doing these experiments, we put the drugs directly onto the cell culture, directly into their medium. So that means we have direct access right to the cells. And again, in the body, therapeutically, that just doesn't happen. You know, when we give a medicine, if we take it orally or it gets injected, moves around in our bloodstream, it's got to diffuse through tissues and get to the tumor site. And so there are lots of compounds out there that when you add them directly to a cancer cell, you might be able to kill the cancer cell, but that doesn't necessarily mean that when you go into a whole body that you will be able to get the drug to the tumor, and that you'll be able to have the same effect in a ball of cells. But this is where we start when we're doing experiments like this. If you find that your compound works in this kind of situation, then you escalate. You move then into in vivo models. You might move into different ways of testing this more physiologically, and then you move into man. So we have a lot of data about cannabinoids in this kind of setting where cannabinoids are added directly to cancer cells, effectively in like a petri dish type scenario. And as I've mentioned, almost every cannabinoid that has been tested has been found to inhibit the growth of these cells, of cancer cells specifically. Cancer cells appear to be quite intolerant to cannabinoids. They inhibit the growth, and they also kill if the cells are already grown. So if your plate is already covered in these little cells, like in that picture there, then they will kill them. They will either cause programmed cell death, which is called apoptosis, or they will cause necrosis, which is just like killing a cell normally. There's been a lot of research that has looked at how this happens, and multiple receptors are involved, and this will depend on the cannabinoid molecule that you're using and on the cancer. But some of the typical receptors are CB1, CB2, the TRP channels, and the PPAR nuclear receptors. They are the main mechanisms by which cannabinoids bring about cytotoxicity in cancer cells. And one thing to point out is that we do require quite high concentrations of cannabinoids, even in an in vitro unphysiological setting like this, where we're adding the cannabinoids right onto the cancer cells. We do need quite a lot of compound, far more than would be circulating in your blood if you were taking cannabinoids. And that's something that's really worth bearing in mind, is that they're not very potent. Although they do it, they're effective at what they do. They're not very potent drugs. You do need quite a lot of the drug there. You also need time. So a lot of these compounds take several days to kill the cancer cells. It's not a quick reaction. It takes days for the cancer cell to go, okay, I don't like this, I'm going to die now. So it takes time, and it takes quite a lot of the drug to kill the cancer cells in vitro. And this is the kind of data that we get when we do these kind of experiments. I've done plenty of these in the past. You get concentration response curves, and you can really play around. You can do quite a lot of work. You can look at a combination of drugs. You can see, for example, these researchers have done different combinations of CBD plus THC, or CBD plus CBG, CBG plus THC. So in vitro experiments, because they're fairly quick and easy to do, they give you the ability to really investigate how they work, use receptor antagonists, figure out what the mechanism of action is, look at different compounds, see which ones are more potent, which ones are more effective, what happens if I add different compounds in different ratios. And so you can learn a lot through these types of in vitro experiments. I don't want to completely put them down, because, as I said, this is something we do a lot of, and it is really your start point when you're doing this kind of research. So when you are happy that the compound that you're investigating kills the cancer cells that you are interested in, let's say we're in prostate cancer, for example, and you've done quite a lot of work, you figured out what the concentrations are that is required to kill them, you figured out how they work, and whether maybe you might do some work to see whether they work better in combination or better alone. And then you might move into an animal model. And the type of animal models, just again, so that you can really visualize what we mean when we say that cannabinoids reduce tumor size in animals. What experimenters will do is they will grow cancer cells in these in vitro, in petri dishes. They will grow them up, and then they will inject them into the animal model, and it will normally be quite an immunosuppressed animal, just to allow the tumor cells to take. They will be put under the skin, or potentially injected straight to the site that you want to induce a tumor in. So, for example, with prostate cancer models, you can put the prostate cancer cells under the skin and have the tumor grow there under the skin, because then you can easily measure it. Or you could inject those cancer cells straight into the prostate of the animal and have the tumor grow where it should do, in the right location. And people use both kind of varieties of cancer models. And then the drugs are normally delivered IP, which is intraperitoneally. And by drugs, I mean the cannabinoids, which is an injection into their stomach. And the reason I highlight this is because, again, this is not very therapeutically relevant. In a human, we tend to give drugs either IV or through an oral delivery route. We don't tend to do IP injection, and actually it can have a big impact on the doses that's required, because you usually need less of a drug if you're injecting it straight into the peritoneum than you do if you're giving it orally, because when you give it orally, it has to go through your digestive tract and be absorbed, and has to go through the liver and be metabolized. So cannabinoids work in these type of models. So there's an awful lot of work that has been done and found that various different cannabinoids in various different types of cancer models have found that cannabinoids reduce tumor size in animal work. And typically the kind of effective doses that we need when we go into an animal model are between 5 and 10 milligrams per kilogram per day. So that's the type of doses that is required, at least in a mouse model.